Hello, neuro class. So I'm going to talk a little bit about these three GABA papers that we're focusing on Tuesday. So I'm going to try to summarize them in the order that they are, starting with the Kravitz paper. They're all pretty old papers, which makes them a little bit easier to read and makes the research really interesting because it was at the forefront of its time. So this first paper is the Kravitz paper. It's the 1960 paper, and it was one of the first papers of its time to really show that GABA is involved in inhibitory neurons and that GABA is likely an inhibitory neurotransmitter. And this came at a time when the doctrine was that GABA wasn't involved in inhibitory neurotransmitters at all, and Dr. Kravitz actually kind of went against this grain and sought to reevaluate the data and this discovery. And good thing he did. So this paper starts off, it's again using lobsters. So they use lobster neurons to determine that GABA was present in inhibitory neurons and not excitatory neurons. And they take a region of the second claw in the lobster and they basically open it up, and there's only two neurons in this region. There's an excitatory and an inhibitory neuron, or a motor, they call it in this paper, and an inhibitory neuron. And they separate those two neurons and they stimulate them. And based on their response on the innervated muscle, they can determine whether they're excitatory or inhibitory, and they separate them. After separating these two neurons, they homogenize the neurons. And they basically take the components of this neuron and run them through an enzyme assay test measuring for GABA activity. And so there's a figure in your paper that looks at this, and I would take a look at the diagram. It's in the discussion of this first Kravitz paper, and it basically shows you how they enzymatically did this. And so they're looking for the production of NADPH. And yeah, you can take a look at some of the enzymes involved in that. So they weren't measuring production of GABA, they were measuring production of one of GABA's side products with one of the other enzymes in the reaction. And so they're measuring for the production of NADPH in this, and they showed basically in these first two tables that GABA is primarily involved in inhibitory neurotransmitters, not the motor neurons, or not the excitatory neurons. So yeah, that was the major take home of this paper, that inhibitory neurons primarily have GABA in them, whereas excitatory neurons had basically no GABA present. And so they also verified this using chromatography. And so they basically did, I don't know if anyone of you remember thin layer chromatography from organic chemistry, but that's basically the other method they used to evaluate if GABA was present in these neurons. So by basically comparing a known standard line on the chromatography paper, so they knew where GABA should be located, they knew where some of the other inhibitory neurotransmitters should be located, they ran these thin layer chromatography with inhibitory and excitatory neurons and the components of each. And in the inhibitory neurotransmitters, or inhibitory neurons, they saw where the GABA spot was in conjunction with where it should have been based on the standard, and in the excitatory neurons they didn't see any GABA present on the thin layer chromatography. So the next paper that kind of follows a sequence of papers is the Otsuko et al. Paper, and here the researchers were trying to look at if they could prove that GABA was actually released from inhibitory neurons and not from excitatory neurons. And so they did so without going too much into the methods of this, by basically collecting all the GABA that's produced in the neuromuscular junction and then measuring the amount of GABA and how much is present. And they found that inhibitory neurons had a significant amount more GABA present than excitatory neurons, which was expected. That was the hypothesis. And they were also able to basically remove the calcium from the axoplasm, and this was to prove kind of that GABA is a neurotransmitter. And by removing calcium, they could basically inhibit GABA release. So that was really interesting because it suggested that yes, GABA is definitely involved in inhibitory nerves, and it's also a neurotransmitter because calcium is responsible for releasing it. And in this paper, they also showed that the stimulus frequency and intensity when they stimulated these neurons to release GABA was directly responsible for how much GABA was produced in these neurons. So a big take home of this second paper was that they could stimulate excitatory and inhibitory neurons, and only the inhibitory neurons really release GABA in a measurable amount. And when they removed calcium from the axoplasm here, they could basically stop the release of GABA or release of this neurotransmitter. So in this last paper, the Hall 1970 paper, the researchers were primarily interested in determining the mechanism by which GABA is produced in inhibitory neurons. So in the first two, we looked at, well, is GABA present in inhibitory neurons? Then we looked at to see if it was released, and now we're looking to see at how specifically it's produced in a unique way compared to excitatory neurons. So to do so, the researchers first used radioactively labeled GABA to measure how quickly it was produced in comparison between inhibitory and excitatory neurons, and here they saw that GABA was basically produced only in inhibitory neurons. The researchers then looked at several important enzymes involved in the GABA synthesis from glutamate and found that glutamate decarboxylase, so removing a CO2 group basically from glutamate, was the primary enzyme that was active in inhibitory neurons and not really excitatory neurons. There were several other enzymes that were present and acting in both, and this was the one that sort of stood out as being different. So they measured this by looking at the amount of CO2 that was produced during the reactions. And again, these reactions are shown in the figure in paper 3 and paper 1. And this indicates that the ability to produce GABA in inhibitory neurons is directly linked to the ability to accumulate this enzyme glutamate decarboxylase. I hope this video helped a little bit and helps kind of give you an idea of where to go and how to look at these papers. They're all pretty exciting and pretty interesting, and they have really historical importance associated with them. Thanks.